Good evening. Thank you for joining this online class on bioinformatics training. Within a short notice, you joined the meeting. Thank you again. Today we shall discuss on metagenomics, next generation sequencing, and obviously, how can you utilize this emerging tool for solving new biological question and also discover new knowledge useful for sustainable agriculture. I shall try to share some of our experiences as well as the basic concept, uh, the scope and importance, and obviously the technique we used. Uh, basically, we applied metagenomics for several experiment. One of them was gut microbiome of Hilsha, microbiome of microbiome diversity in Bangladeshi soils, and also microbiome diversity in the rhizosphere. When we bioengineer, how can we manipulate for the benefit of the plant productivity? So let me show you some of the slides. Mm, Just a moment. I'll change this one and show you another. Yeah. Is the slide visible?
No, sir.
No. Okay.
Not now, sir.
Okay. I shall share again. I guess now it is visible, right? Is it visible now?
Yes, sir.
Okay. Yeah. So today we shall discuss metagenomics and its application for understanding the microbiome uh, in the environment, including uh, different ecosystem, even any organisms, guards, or even the soil, cultivated soil, or anything, any environmental samples. In our previous class, we discussed a bit on metagenomics. How can we define metagenomics? The genomes of total microorganisms or microbiota found in nature, representing the genetic diversity of uncultured microbes that represent about 99%. So metagenome means the total genomes of all organisms living uh, in any environmental samples. And previously, I shared with you that a large proportion of the environmental microorganisms are unculturable. A large proportion means about 99%. So, in the culture media, over the years, microbiologists tried to simulate the real environment for the growth and reproduction of different microorganisms, but our success rate is about 1%. So, metagenome means the genetic codes of all organisms that are uh, living in an interacting uh, situation is very important to understand for their uh, practical application. Metagenome from biomass degradable environments like activity screening and sequencing, bacterial biodiversity by next generation sequencing. Some of the works have already been done, but this is still an emerging area. The most important application of metagenome and importance being realized when human microbiome project was led by Cambridge University and uh, surprisingly we understood that an adult human carry uh, 3 to 6 pounds of microorganism. Uh, it's really incredible amount and total number of cells of microorganism in an adult, healthy human is about tenfold higher than the cells he or she has. Really incredible in numbers, but as they are microscopic, we cannot see them. And a recent study confirmed that uh, these uh, microbiome, the microbiome in human uh, are about uh, more than 150 species or even more, and they uh, play a vital role in the fitness of human health, growth, reproduction, and even uh, they are considered as second brain, especially the gut microbiome of human. So, compared to if we think about other eukaryotes like plants or animals, even the soil, the role of the microbiome. Disconnected, right? I'm sorry. Am I audible at this moment? And can you see the slide clearly?
Yes, sir, you are audible and the slide is being shared.
Okay, thank you so much. But somehow, uh, you know, uh, it was disconnected. I'm not sure whether recording is still okay. Let me see. If recorded, then maybe you can. Yes. Unfortunately, the internet is disturbing today. But now I'm not sure when I was disconnected, but I was talking about the importance of human microbiome and gut microbiome of human is considered the second brain because of many neurons are available in the gut and they are uh, functioning like the brain. Historically, metagenomic technology uh, significantly progressed over the years and the price is dramatically decreased and we are now getting huge uh, the data in the gene bank and mostly people are focusing the bacterium that is all the bacteria present in the guts of human and other animals even environmental samples but recent progress in the next generation sequencing, that is shotgun metagenomics, we can capture the whole genome of all organisms present in an environmental samples. And the technology is progressing very fast. Total market of metagenomics is, $1 billion nowadays, and it is expanding steadily. The purpose of metagenomics, already I gave you an example. The most important job is to know who is there, that is in the environmental samples, plant tissue, rhizosphere or gut of any organism, even soil sample, uh, water sample. We can ask the question who is there, that is who is means what microorganisms are there, because the large organism we can see, and the biodiversity of visible organisms, plants, flora and fauna, people are studying, and huge data are available. But microorganisms, uh, the studies are still limited. Our understanding is very limited, but they are the vital forces uh, that regulate the geochemical nutrient cycling in the environment. And not only that, the climate change or global warming, that is, atmospheric gases and the elements present in the soil, they are mostly uh, influenced by the function of those microorganisms. This is why their functions are very important. Another issue is the genes present in diverse microorganisms. They are very important for uh, using them for the production of new biologicals, new enzyme, new metabolites to solve the health issues and other problems. So very very important, uh, untapped natural reservoir of bioresources. So now this area is highly focused. I shall tell you later more details. And what are they doing? As I mentioned, uh, in the group of microbiome in the environment, they can be prokaryotic or eukaryotic. For example, fungi are eukaryotic, nematodes are eukaryotic, bacteria, archaea, they are prokaryotic. And they have like, uh, large animals or organisms, they have the uh, metabolic pathways. Some of them are uh, very powerful in adapting to the very harsh environment. Uh, so if we can get their genetic code and genes involved for making valuable metabolites, or genes involved in adaptation to the harsh environment, we can easily clone them and use for the human benefit. And not only this simple advantage, but also we can get their stories for uh, utilizing, for understanding the evolutionary biology of the living organism biomes on this earth. How can we do that job? The basic workflow, because if you want to know the microbiome at the uh, sea beach of the Bay of Bengal, for example, in Cox's Bazar, how can you start? First, you need to collect the sample. Sample can be water, sample can be soil, sample can be even plant tissue or animal tissue, um, depending on your target. So you collect the samples. During the collection of samples, you need to be careful about uh, the contamination. Contamination means you need to collect the samples from any ecological area or ecosystem with gloves and mask and put the sample in uh, the ice box and bring to the laboratory and then extract the DNA. If it takes time to take extraction, you need to put the sorry sample in minus 80 degrees Celsius. Then all the organisms uh, they will be inactive, but DNA will remain intact. So you can extract the DNA. To extract the DNA, you can use several methods are available to extract the DNA. DNA, locates in the nucleus on the chromosome. Uh, we called chromosomal DNA or nuclear DNA. DNA also locates in the mitochondria, and in case of plants, DNA also locates in the plastids. Small amount in plastids, small amount in mitochondria, but uh, major amount of DNA locates on the chromosome. So you need to crush the cell. In that case, we use liquid nitrogen to crush the cell and get the DNA in the buffer solution. And there is the steps. You can get the commercial kit. They have the instruction. How can we get the DNA and purify it? And then you need to uh, measure the concentration. A certain level of concentration is important for getting the or trapping the all organisms. Then there is an amplification process. For example, if you want to do 16S rRNA gene sequence for understanding the bacterium, that is all bacteria present in your sample, then you can do the PCR using the specific primer. For fungi, you can use the sorry the ITS region, the conserved region. But in case of shotgun metagenomics, sometimes after collecting the sample, we do the enrichment, that is in the media, allow them to uh, improve the quantity. But it can increase the number of population. But in the laboratory condition, we cannot increase the number of all the organisms. But anyway, the DNA concentration is important, and then you go for the analysis. Analysis means sequencing, sequencing, and after sequencing, bioinformatic analysis. So this is a flowchart. And microbiome, I already defined the community of microorganisms that can usually be found living together in any given habitat. Maybe, for example, in our gut or at the rhizosphere or even in the fish gut or fish skin, like that. So microbiome is the subject of a study since 1952 when the Scientific Monthly, have proposed this, term. And then it was defined more precisely, the microbiome, in 1988 by Whipps and co-workers, a characteristic microbial community occupying a reasonable, well-defined habitat which has distinct physicochemical properties. So this term is now uh, very popular in microbiology and does not only refers the microorganisms involved, but also encompasses their theater of activity. Theater activity. In the theater, every player has some activity. In the ecosystem, every microorganism has a defined role, and ultimately their crosstalks result the uh, ecological balance. So uh, it is termed as theater of activity. So here you can briefly see what are present in the microbiome: bacteria, archaea, fungi, protists, algae, and many more microorganisms, even virus also. Theater of activity means microbial structural components. Uh, they are cell wall or cellular structure, proteins, peptides, lipids, polysaccharides, and the DNA sequence and different, for example, plasmid, mobile DNA or like that. And also they can produce lots of metabolites. Some of them produce toxins, and obviously they produce diverse signaling molecules. Signaling molecules is the molecule of language to talk intra-specific or interspecific. And all the stories are really exciting to know for getting the uh, benefit for the mankind. Uh, and we call biome a reasonable, well-defined habitat which has distinct biophysical properties. So all biome, how they act each other, interact with each other, and impact on the environment, and also the dynamics of the climate change, uh, the cumulative interactions can explain. And on the world, the herbivores uh, are the dominant uh, members. Herbivores means those are live on green plants, and there are carnivores also, herbivores, human also herbivore. So there are lots of herbivores on this earth, and herbivores also harbor lots of microorganisms. Here you can see the how they are different kind of phylum of microorganisms, and here you can see herbivore harbors more diverse microorganisms than the carnivore, and they are interacting with each other. And another interesting point is the holobiont evolution. Human, plant, and all other organisms, they are interconnected. And some of them are commensal, no harm or no benefit or like that. Some of them are symbiont. So the relations, some are pathogens. So in relationship of or organisms in a given ecosystem is incredibly diverse. So I would like to introduce you another term, hologenome. A holobiont is an assemblage of a host and the many other species living in or around it. For example, uh, the assemblage of all living microorganisms inside our body or tissue or surface or around us, uh, holobiont of human. And they together form a discrete ecological unit through symbiosis, through though there is a controversy over this discreteness, because not only symbiosis, there are some antagonistic relationship also. The component of a holobiont are individual species or biomes, which while the combined genome of all biomes is the hologenome. Very interestingly, hologenome in a given system, for example, a pond, if you analyze the uh, microbiome or whole genome of a fish, you can find diverse microorganisms. And the prokaryote eukaryote or the vertebrate fish. But if you analyze the genome, you can see a significant portion is shared with each other. So really exciting, and through horizontal gene transfer over the evolutionary process, it happens. For example, human microbiome contributed nearly 30% of the gene present in the human DNA. Really exciting. We achieved, and we are carrying 70% of the genome from the, primate, but 30% nearly from the microbiome uh, through the evolutionary process and horizontal gene transfer. So, understanding the whole genome uh, really exciting to engineer them for the benefit of the host, maybe plant, fish, animal, or human. Here you can see this is the how can we uh, progress. Previously, I mentioned who is there, what can they do, what are they doing. So uh, previously people used a microscopic or uh, culturomics, that is culture characterization of cellular function based on the culture, but very limited story. But now we can extract the DNA, RNA, protein metabolites, so we can do metagenomics, metatranscriptomics, metaproteomics, and metabolomics, and get more story. And the emergence of next generation sequencing and the progress of next generation sequencing and computational methods allowing us to generate incredibly large amount of data, and those big data are the big asset but very complex to handle. In case of agriculture, the uh, soil rhizosphere, that is below ground and above ground, lots of microbes are associated with the plant, and they are playing vital role in uh, adapting of a plant to the adverse environment. Nowadays, people are very much interested how weeds, without any care, non-domesticated, that is, weed may be unwanted plant, they survive in the harsh environment without any care and grow very fast. So, what kind of microbes are over there? One of our student Kanis, she analyzed and discovered many weeds that can improve the health of the cultivated plant also. So soil microbiome uh, is not only discovering the diversity and their function. Now a new topic, microbiome engineering, not only in case of soil. In other cases, gut microbiome engineering is becoming a powerful tool. Here you can see a review article published in Nature Biotechnology. Microbiome engineering means you somehow change the microbiome of an ecosystem. And how can you, it can be done just, adding, manipulating the beneficial microbes uh, that are culturable or even non-culturable uh, in an ecosystem, even in the gut. Then allowed a new interacting system, interactome. As a result, due to interactions, some will, a new, uh, homeostasis will be developed. So the initial, the microbiome diversity and population would be changed. And if this change is beneficial for you, then that would be very good. And in case of environment, we can also do similar engineering. For example, unfertile soil or highly acidic soil or alkaline soil or saline soil. By using microbiome engineering, we can make them productive soil. So microbiome engineering, here you can see, uh, you understand the microbiome and find some of the microbiome that are beneficial for you, and then you can apply them to change the soil microbiome diversity, and by manipulating and adding new microbiome, and there would be the whole ecological balance among the interaction would be changed, and it is called microbiome engineering. And people are doing, and especially the positive uh, role playing microbes you can add. Lots of literatures are present. People are now using the modern tools for mapping the soil microbiome. In Bangladesh, we have soil maps for each of the upazila physico-chemical properties, but soil definition of a soil is a living entity. So microbes are the life of the soil. So, without understanding the microbiome, only the physicochemical properties, those are basically regulated by the microbes, is the part of the story of our soil science. Now, mapping of microbiome, like GIS mapping for arsenic or salinity, you can find the or soil acidity. Microbiome mapping is an emerging area. In Bangladesh, it has not yet been done, but many places in the world, even in Europe, America, they have the microbiome mapping. And based on the microbiome mapping, you can engineer the microbiome if the microbiome is not productive for the useful for the crop. So I'm not giving more complex issues, but mapping African soil microbiome is a new project, and it is funded by the uh, Bill and Melinda Gates Foundation and USAID. So in Africa, they are doing the microbiome mapping, whole Africa. But in Bangladesh, we have not yet done. Recently, we started this project. So mapping microbiome uh, would allow us to understand the microbial diversity and the population, and find the new signature or marker for understanding the productivity of the soil based on the particular microorganisms there. So microbiome mapping in Bangladesh, I think, very important. And we have a modest start. Soon we shall publish the microbiome diversity in several 14 agroecological zones, and others are underway. Here, the primary work of microbiome and soil pH. In our study, we found that some soils, in Bangladesh are acidic, and this is due to uh, the organic acids and also mineralization process done by the microorganisms. So people are using the lime and other things, but never care about the microorganism there. But microbiome engineering can solve this problem. So these are some data. I will give you the slide you can go through. So in case of microbiome or metagenome sequencing, I mentioned you, we can target the organism, maybe fraction of the story like 16S rRNA gene sequencing to get the bacterial diversity or bacterium. ITS we use for the fungal diversity or fungi only, but shotgun or whole metagenome sequencing we can do for understanding all the microorganisms present there and get the whole story. But it is very complex to analyze the data. Uh, and day by day, bioinformatics tools are becoming easier. For example, in case of 16S rRNA that target the uh, bacterial particular gene to understand the community composition. But in case of shotgun metagenomics, we can get uh, the whole microbiome diversity. The flow chart is here. So if you have only one sample, for example, our farm sample, experimental farm sample, then you can get the operational taxonomic unit. That is, it is called a group of closely related individuals which are arranged together based on the similarity of specific sequences. So we can easily get the operational taxonomic units, how many, uh, you know, closely related organisms are there. There are lots of, bioinformatic analysis after having the data, but this workflow is very important. I think the uh, metagenomics or 16S rRNA gene sequencing, metaproteomics, metatranscriptomics, bolomics or genomics, all things we can do to get the, who are there, what are they doing, and who are related to whom, and their overall, function to the ecosystem and the environment. So quite complex, but the methods are progressing. So we can even get the information at the protein level or metabolome level from the shotgun metagenomics analysis. So here I am also giving again the uh, after the first you need to get the sample and then sequencing, and after that analyze the sequence data by using different bioinformatic tool and targeting what you want to analyze and what kind of question you want to solve. Here is another flow chart. You can see how we can do the 16S or the shotgun metagenomics to get the uh, whole story. Here, one of our article, Dr. Nazmul from Veterinary and Animal Science faculty, he published, and I was also involved in this work, published in Animal Microbiome. What he did here, induction of mastitis by cow to mouse fecal and milk microbiota transplantation causes microbiome dysbiosis and genomic functional perturbation in mice. Uh, Mastitis is a disease caused by the several microorganisms, not the single one, and complex group of bacteria in the udder of the uh, dairy cows. And it is really a big problem for the dairy industry. If mastitis is severe, then milk production dramatically decreased. Even blood comes from with the milk. So Nazmul studied about the mastitis, how and the mastitis causing microorganisms interact with each other. And if mastitis occur, then what happens in the microbiome of the cow? So he used the mouse model. So what he did, he used milk from the infected uh, a cow and also fecal samples and transplanted to the mice and analyze the mice is a very good model for the mammals, how it affect the microbiome of the mice and how it affect the uh, you know health parameters. And it was a fantastic study. We also it was a shotgun metagenomic study and robust study. You can go through this paper published in Animal Microbiome. Another work we did during the SARS-CoV-2 viral, pandemic causing by SARS-CoV-2 virus. And we collected the samples from the nasopharyngeal tract, and we found that when infection occurs, the microbiome of human uh, change, and especially pathobionts, the microbes causing the disease, especially airborne and opportunistic pathogens. They recruit not only the SARS-CoV-2 effect, other pathobionts join and intensify the problem. This is and which microbes recruit after the immunocompromised uh, human due to the COVID infection, SARS-CoV-2 infection. We, figure out, and it would help to, uh, suggest what kind of drug is this needed. Today I shall show you a bit detail of this work. Possibly you have got the news that gut microbiome diversity and distribution in the national fish hilsa, Teno Elusa Elisha of Bangladesh was done by our group. And why we did this job? Uh, we understood that you know that the hilsa is uh, a powerful fish, our national fish. Uh, Delicacy of hilsa is unique compared to any other fish. It lives in the Bay of Bengal in the marine water, but it travels to the brackish water as well as the sweet water, fresh water during the spawning season, that is for spawning. And our rivers are polluted with, heavy metals, microplastic, and lots of other things. But even though, and lots of microbes are also there in our country, aquaculture is threatened by the different diseases. But in case of Hilsha, in last 100 years, there is no report on the occurrence of any disease. So we thought gut microbiome of Hilsha might play a role for the fitness of this fish. And this fish is the most expensive fish in the world. But you see, Hilsha, if you take out from the water within a few minutes, it dies. Uh, but in water, it is very powerful. The speed, actually, I don't know how it moves faster. So we thought Hilsha might contain some of the microorganisms. Especially, we first targeted the bacteria. Among the microorganisms, bacteria and archaea very powerful in as a gut microbiome. And interestingly, we found several genera of the bacteria uh, found in the gut microbiome of Hilsha have not been reported in any other fish gut. They might have the probiotic function for the fitness of the Hilsha. Probiotic means the gut microbiome or microbiome present in any host that have a positive role on the health and fitness of the host. So probiotic is now a big industry. Human probiotics you can get as a capsule, and probiotic treatment is becoming very popular. In case of fish aquaculture, uh, in shrimp industry, we import a large amount of probiotics from other country. But we thought Hilshagat might be a gold mine for uh, probiotics. This is why we did the job. So how we did the job? First, we collected the hilsa from different habitat. Hilsa uh, uh, fish lives in the marine water, like in the Cox's Bay of Bengal. We collected the marine samples from the marine Cox's Bazar, and then we collected samples from the Chatpu, Rajshahi, that means Bodda, or many other places where Hilsha are, abundance are higher. That means Hilsha from the marine, brackish, as well as the fresh water. To understand uh, the presence of the microbiome diversity, because the pH, salinity, and other things, even temperature, quite different from the upstream river and the downstream Bay of Bengal. How uh, uh, these changes are, tolerated by the Hilsha, or how Hilsha adapted to those environmental changes. We had a special interest. So first we extracted the DNA, genomic DNA, and then prepared the library. Previously, I taught you how library preparation is done. That is, by using enzyme, we cut the pieces and then use a different adapter to both sides of the piece for doing the sequencing. And after sequencing, we did the bioinformatic analysis using some common tools for analyzing the bacterial diversity who are there. I shall show you later on, and also use the R package for statistical analysis. Uh, Here you can see the bacterium diversity in Hillshire, the habitat, fresh water, marine water, or the river water. We found the alpha diversity. Alpha diversity means within the group, for example, uh, group from the habitat that is fresh water, and then brackish water, and marine water. Quite diverse. That means when Hilsha moves from marine environment to the brackish environment and then moves to the fresh water, microbiome diversity changes. This is really amazing. And how it happens, we do not know. That means this changes, changing environment, they, carry changed bacteria also. And we collected the bacteria, studied the bacteria in the gut, not the surface. And we also got the, diversity within the species. There are some common, bacteria in all the habitat uh, of the hillsha, but they are core. But depending on the habitat, some of them are changed. Here you can see marine water, brackish water, and fresh water, the changes of different uh, taxonomic profile, that is phylum level, bacterial, different phylum, you can find quite different. Uh, so you can go through. And taxonomic profile of bacterium in Hilsha at the order level. These are the different orders present there. So we found some of the orders are completely new in Hilsha gut, not in other organisms. We also moved to the bacterium at the genus level, even move to the species level. And if you see the genus level, fresh water, brackish water, and marine water, the colors, you can quickly check why different than the one group to another group. And we analyzed the function of this genus available in the literature. And we found them, some of them are associated with the salinity tolerance or heavy metal tolerance or pollution tolerance or temperature tolerance. So we are not sure how they are helping Hilsha to uh, be smart and fit to the changing environment. A really interesting area of further research. We are now going to do samples are now in UK for shotgun metagenomics, and Rabiul Kausar, our teacher, of fishes faculty. He's doing PhD at University of Nottingham, and he went with this idea of my research and doing job over there. We even analyzed a particular genus in different habitat, and we found that some genus are significantly different in particular, habitat of the hillshark. Uh, so obviously, we found that some of the genus they are potential probiotic genera because they are already known. The genera have the probiotic properties, that is to help the fitness of and disease resistance and tolerance to the environmental changes of the host. Uh, so these are really interesting. And another is some of the genus we identified for the first time in the Hilshagat, not in any other marine fishes. So this is one of the story. If you are interested, please go through the paper published in PLOS ONE. You can get the methods and data also available in the NCBI Gene Bank. You can analyze the data using the tools mentioned over there. And if you need if you want to regenerate the analyze data, um, obviously, I shall advise you to meet with me or discuss with Dr. Nazmul because he did large of the analysis. We also did another experiment. Uh, you know, some of the plant growth promoting bacteria that can improve the growth and yield of the uh, rice or other plants. We discovered two uh, bacteria, one Parabarcholderia, another Delphia Parabarcholderia fungorum, and Delphia surhatensis from the as endophyte. They can improve the plant growth, rice growth and yield, and reduce 50% of the NPK fertilizer. But our interest was when we apply them in the rhizosphere as root colonization in the natural environment, how they do perform the job. And interestingly, we found that after few weeks, their population dramatically decreased, although we manipulated. But overall, the rhizosphere microbiome dramatically being changed. So this is really interesting study how the rhizosphere and root microbiome total microbiome diversity and population are changed if you apply a certain bacteria at high concentration. That means it is one kind of microbiome engineering. So we started this work, and this paper published last year, but already been cited 40 times or like that. So here you can see how the application of to bacteria change the microbiome diversity. As I mentioned before, SARS-CoV-2 infection, uh, we analyzed this paper. You can visit the scientific reports, Nature Group's journal to get the data. Not only, microbiome can, interact with each other, they can even regulate the expression of the host gene. This is really amazing. Not direct, indirectly, expression of the host gene can be regulate or managed by the application of microbiome. In case of SARS-CoV-2 infection, we found that host gene expression associated with different pathways are overexpressed, and that was associated with the damage caused by the SARS-CoV-2 disease. For example, pathway associated with the sugar metabolism or kidney function or liver function. Uh, if dysregulated, then you can complexity of the microorganism you can see. So the microbiome diversity analysis, functional analysis, and they are they connect with each other or interact to is really an amazing area for the bio expert in computational biology and bioinformatics. Uh, all the bioinformatics tools, advanced tools, are now focusing to understand these complex interactions and make the inference that is conclusion and address the new challenges ahead in agriculture or in the living organisms. Some of the microorganisms even the signature or marker of particular disease. So microbiome can even, for example, some of the uh, microbiome are associated with specific cancer development, cancer cell development in the human. So you can simply trace that microbe in the blood, and you can early detect the uh, disease. So we are thinking microbiome might be uh, a marker for the soil fertility and productivity. If we can establish this idea, then no need to do whole soil analysis and spend huge money. Just trace some of the signature microbiome present in the soil to understand the soil health. So this is all about 1 hour lecture. You can now ask some of the question. Please ask freely some of the questions so that we can discuss more. Just unmute yourself and then ask the question. Hello. Anyone can start asking the question. In total, 12 participants I can see. Shahriar? Oh, Rubayat. Yes, please ask the question. Or make the question, comment, share your thoughts so that we can have uh, an interactive session. Rubaya? No. Shahriar?
Sir, I don't have any particular question, sir.
Okay. So, what is your sense on this lecture? Is it interesting or beneficial for you?
Yes, sir. The total metagenomic topic is very interesting for me, and I'm very much interested to work on metagenomic in future, especially in related to eDNA to identify fish abundance using the genetic data.
Excellent. Can you explain your understanding of eDNA, that is environmental DNA?
Sir.
What is the modern trend to trace the eDNA?
So, as far as I know, mainly water samples are collected from. It can be a natural water body, or mainly to identify the abundance, fish abundance there, and what type of species are present there. So
Excellent.
they and they
Yeah,
filtered the.
yeah. For the understanding the biodiversity of fishes, uh, eDNA, you think, is an emerging, tool rather than catching
Yes,
all
sir.
the fishes
Yes, sir.
and, count their. Yeah, really interesting. So, how did you got this idea, eDNA?
Uh, Yes, sir, I was really very much interested in doing metagenomic data analysis. So I look for what are the scope of this data and what we do with this data. So I saw some of the work that are rather than sampling fish with net. Uh, some researchers used eDNA technique to identify the variation in the species and their abundance. So it really intrigued me. We can use this technique in our various natural bodies, such as in the sillet region or in the brackish water region or in the even in the marine region also.
Excellent. That means in any ecosystem or habitat, if you get the environmental sample, you can trace the presence of all living organisms. You are interested in case of fish, even other organisms, that is total biodiversity based on the environmental DNA. For example, if you want to do the biodiversity of Sundarban, one of our colleague, she did PhD in Germany, now doing job at veterinary animal science faculty. She visited me and she told me only, collecting the flies in the Sundarban at different locations and then extract the DNA from the flies, uh, you can get the all living organisms over there. Because flies, land on different organisms present in the environment during the, flight. So they get the DNA from diverse organisms, and flies are, champion traveler. So from the flies, you can trace the, that is, uh, fingerprint the diversity of all organisms over there. So this is really a power of environmental DNA. Thank you so much, Shariar. You added a new topic here, environmental DNA. Environmental DNA includes, uh, uh, you know, uh, trace DNA, uh, not only from the living cells, even your DNA in your clothes, or even the DNA in the you used any items that you touch can be traced nowadays by using metagenomics. So organism present in an ecosystem we can trace by using eDNA. But metagenomics we can do um, for tracing microorganisms that are really even at higher depth of sequencing using shotgun metagenomics can give us a huge potential for their practical application and discovering new biologicals for AMR resistant superbugs as drug, or even many more new benefits from the uh, enzymes, metabolites produced by different organisms. Thank you so much, Shahriar. Any other? Uh, I'm expecting some more, thoughts and interactions from other students, participants. Ishtiaq, do you have any question, comment? or thought that you like to share here? Or Rubayat, Sudeshna Sarkar, I can see Khaled, Manna Islam. Just unmute yourself and ask the question. Even no question. Please, say something.
Sorry, sir. No questions, sir.
Okay, Rubayat. Are you in the hall?
No, sir. I'm at home, sir.
Okay, thank you. And how about others? You have still a few minutes time
I'm
to.
also at my home, sir.
Oh, where? Where is it?
Sir, I live in Ashkona. It is just opposite of the Haji Camp in the
Okay.
eastern side.
Okay, thank you. Thank you so much. How about others? Some of the students I saw they were connected, but maybe due to internet, instability, they are disconnected. But anyway, thank you so much in this winter night, and good night to you all. And hopefully, uh, uh, we can see, we can have a face-to-face meeting, a lecture next time. And whenever you have a question in mind about bioinformatics, new thought, and please visit me, because the analytical tool maybe a you can get training, but remember that why you use this tool, you must understood or understand well. This is really very, very crucial, because the tools and softwares we are using now, they are fast evolving. And if you know uh, if you have the defined biological question which need to be addressed, if you have a good idea matters, and then you can find the tools that needs to be used. And many of the bioinformatics tools, most of them, you can find the procedure step by step, like cooking a recipe in the YouTube. So whenever you have a nice question and you want to start the bioinformatics analysis, you can get lots of video, recorded video to follow, and you can solve your question. So the tools are just tools, but scientific questions are very, very important. If question is new, then you can discover new knowledge, new information, and then how the information could be utilized for the benefit of human is another area of your thinking. Thank you so much, and have a nice yeah. Any question?
So thank you, sir. Thank you, sir, for the nice session, sir.
Oh, thank you. Good night, and have a nice weekend.
Good night,